Welcome back to Unit 3, Lesson 6. This is part 2 of the video. And now we're going to talk about why and how we sequence DNA. And the problem with this little discussion here is it's very quickly becoming obsolete. The method I'm going to tell you about is in transition right now, and we're actually picking up new methods, faster methods for sequencing DNA. But the concept and how it works is quite important, so we will still learn about it. So the first thing is, why do we sequence DNA? And there's many reasons. So we can look at paternity, ancestry, and crime scenes. So basically, once we've done our PCR, we can then do our sequencing and use both of them for similar purposes. So to sequence DNA, we just slightly alter the process of PCR by replacing our nucleotides with one form of what's called a dideoxy nucleotide that is fluorescent. So by dideoxy, we take our DNA with its phosphate group, and DNA normally has an H here, and the base, that's why it's deoxyribose. This one's dideoxy, and we've got rid of the O here and put just an H, and the base itself, in some way, shape, or form, will be fluorescent. A will be one color, T will be a different color, G a different color, and C a different color. Okay, and by making it dideoxy, any place a dideoxy base incorporates, we cannot add on to the chain. So what we do is we create the polymerase reaction with a mixture of the different fluorescent dideoxy bases and of the normal bases, or normal nucleotides. And at random, sometimes you'll incorporate the normal nucleotide and the chain will continue to grow. And sometimes you'll incorporate the dideoxy nucleotide and the chain will stop growing. And what will eventually happen is you get chains of nucleotides of various sizes. When you run your PCR. And what they will then do is they will run these DNA out on a gel. And the gel acts as a sieve, and as the DNA runs, they run it on the gel by using a positive electrode and a negative electrode. Okay. So the negative electrode will be at one end and the positive, and because DNA has a negative phosphate backbone, it will run away from the negative and towards the positive. But the smaller the fragment of the DNA, the faster it can run through this gel. So this is a tiny fragment, the smallest, and this is the longest chain. And they look at the fragments based on the color of fluorescence on them, and they can tell, okay, this longest chain ends in an A because it fluoresces the color of the A, which typically is taken to be green. G is taken to be a darker color, we'll just say, sorry, black. A is typically green, T is typically red, and C is typically blue. Those are the fluorescent colors they attach. Okay. G is actually fluorescent yellow, but when you're trying to read that on a piece of paper, yellow is really hard to read, so they change it to black on the paper. Okay. So they run out the different segments, and they know the longest segment ends in an A, so that's going to be the last letter of the sequence. And then the shortest, they go backwards, so it's going to be an A, then a C, then a G, then a T, then a C, then a C, G, T, A, so forth. So they're able to, through a little trick of adding a dideoxynucleotide that's fluorescent, they're actually able to sequence the DNA. Okay. Once they sequence the DNA, then they can use it to compare to parents' DNA, ancestry DNA. So are you related to that, mummy or whatever, and crime scene DNA. Okay. The next thing they use PCR for is in something called site-directed mutagenesis. And this is where we intentionally mutate DNA to see how the mutation affects protein function. We may suspect a protein or a part of a protein is important for doing something, and we can mutate it and see how it affects the function. Okay? And the way they use this is they use PCR and a primer that is slightly mutated. So not the perfect complement. To introduce a change in the sequence. So when you do site-directed mutagenesis, not every single copy is going to be mutated, because you're going to start out with your template, which is wild type. And that template's going to be in there for the entire sequencing PCR process. And half the time you'll be copying the template, but most of the time after the first round you're going to be copying the mutated version. So this primer is introducing a mutation. This process is proven invaluable to researchers because it allows us to change proteins in a controlled way, or the coding for proteins in a controlled way. And it was first developed by a scientist called Michael Smith, who actually worked and taught at UBC Vancouver. And because it was a modification on Kary Mullis's PCR methods, but it was really important to researchers' ability to discover and determine how various proteins work, he, along with Kary Mullis, won the Nobel Prize in biochemistry in 1993 for this work. So that's site-directed mutagenesis. I'm just going to stop there, and we'll pick up the last bit in part 3 of the video.